I am requesting my technical team. Please confirm whether my voice is clear.
Yes, sir. Voice is clear, sir.
Okay, thank you, sir. Good evening. Good evening to the Honorable Speaker, respected faculty members, and all the participants. Myself is Sarfraz Anwar, and I'm really happy to welcome you all to the fourth day of the AICTE sponsored Faculty Development Program, that is FDP. I hope you all are learning a lot and really enjoying the season so far. So let's take a quick look at today's schedule. In the first season, from 6 p.m. to 7:30, we will have a talk by our honorable speaker, Professor Cesar de la Fuente, and the topic is machine learning. Then, in the second season, from 7:30 to 9 p.m., there will be a joint talk by Dr. Anupriya Kumar and Dr. Lucas Iberlin, and the topic is cryptic pocket detection using in-hand sampling for CARAS. But there is an important update. There is important update for all the participants. The update is our honorable speaker Professor Cesar de la Fuente will be joining us at 6:30 p.m. I'm repeating. Professor Caesar will join us at 6:30 p.m. That is after about 30 minutes. So, in the meantime, my PhD colleague, Mr. Satya Vivek Jaiswal, will give a short demo of about 15 to 20 minutes, and the topic is pharmacogenomics. But before we begin, let me share a brief and insightful introduction of Mr. Satya Vivek Jaiswal. He is a passionate researcher with deep interest in bioinformatics, molecular biology, and computational sciences. He completed his bachelor's in biotechnology from MIET Meerut and his master's in bioinformatics from the Indian Institute of Information Technology, that is IIT Allahabad, where he is currently pursuing his PhD in bioinformatics under the supervision of Dr. Nidhi Mishra and Dr. Pritish Kumar Bharadwaj. His research mainly focuses on understanding complex biological systems through multi-omics data integration, network biology, and computer-aided drug discovery. So now I would like to hand it over to Mr. Satya Vivek Jaiswal for his demo season. Satya, you can start now.
Thank you, sir, first. Thank you for the introduction. Good evening, participants. We are welcoming you to the session of the network pharmacogenomics. So I'll be sharing the. Am I audible to all? Please, someone confirm me. Am I audible? Okay. So I'll be sharing my screen. Is my screen visible to all?
Yeah,
Right?
you please.
Yeah. Thank you, sir. So today's session will be going on the network pharmacogenomics, the changes to the network-driven therapeutic insights. So let's begin with what is pharmacogenomics. Why do we do pharmacogenomics? So let's begin with the why do we do networks matter in the pharmacogenomics? So traditionally, the pharmacogenomics has focused on this one gene and one drug relationship. Here the single gene, single gene variant might alter the drug of the metabolism or toxicity. So, but biology of the particular gene or disease or drug is work very rarely in isolation. So drug response is arises from the crosstalk between the multiple genes, pathways, and regulatory modules. So that's where the network pharmacogenomics comes in the play. Instead of studying the isolated component, we integrated the genomics data with the interaction network to reveal the how gene collectively influence the therapeutics outcome. These system perspectives allow to identify not just a single biomarker but also the coordinated mechanism that drive the gene or disease that according to drug responses. So next is what is the analytical flow to follow this how to do network pharmacogenomics. So these are the major 5 steps that are involved in this analysis. So first we begin with the genomic and transcriptomic data which is we will get from the RNA-seq, microarray and GWAS analysis. Next we will do we'll construct the network in string database, and after building the network we will export the data like network data into the cytoscape and perform the further downstream processing like M code analysis, that is the cluster analysis, identifying the hub genes which are the GB gene is the most important for particular rare disease. And after that we will perform the Klugo analysis that will give you the functional annotation of the particular genes involved in that. To start with the flow, I will going to start the demo session for that. I have the gene set of the I have the gene set, actually the differential gene express data. This is the file. This is the differential gene expressed file, which contains the here the blue part shows that these are the upregulated data, upregulated gene, and at the bottom of that in the red color it is the downregulated. We will differentiate it on the basis of we will divide the data on the basis of log to fold chain, like which gene is expressed in here, highly expressed and in as a upregulated and low expressed as a downregulated. So we will copy. I will start the demonstration here. So first we will start to copy this gene data, and we have to paste it into the string database. I already opened that. So in the string database, we have to choose the multiple protein part, not the single protein name. Multiple protein, and we will paste the all of the gene data, which is it contains the both upregulated and downregulated data. And in the organism section, we will choose Homo sapiens because we are working on the Homo sapiens human data. Okay, human genomic data, and it will open the panel which contain the all of the information of the like genes name and its relation, that is like a related information. And after that we will go for the next step. It will construct a 3D or 2D network of it. So wait, here these are the genes which are interconnected, which is it will construct the network from these genes, and these connection between these genes shows that they are somehow connected with each other in some manner. And at the bottom of that there is a multiple option available, that is reverse lasers and setting. In settings we are going to set the medium confidence, but we can choose according to what our need, the highest confidence and highest median and low confidence. But right now we are choosing the medium confidence interval confidence, and update that. So it will give you the like network which is comes into that range, that confidence interval. And after that we will export the network data in the text file, sorry, tab separated values, tsv file, which can be further used for the cytoscape and cytoscape used in the cytoscape. And this is the cytoscape. We are going to perform the further downstream analysis on this application as a file. First, we are going to import the network from the file, and here these are the string network data, and we will open it in the cytoscape. Here we can see that there is lots of same color genes available in the panel, but and after that we are going to pre-process it for further study. So first we need to know which gene is highly regulated, upregulated or downregulated. For that purpose, we are going to add one more thing. We are going to import the table. Actually, I have shown you these are the table which contain the yeah. These are the gene table which contain the both upregulated and downregulated data, and it also contains the statistics of the like log to fold chain value, adjusted p value, frequency of the gene, and the negative adjusted p value. Okay, so we are going to import this file and merging with the network. So we will perform further we further styling process to get to know which gene is related to which related to upregulated or downregulated. So in file section we are going to import this table from we are going to choose a table from file. So this is the table I was showing previously. So So now it's here. After importing the table, it will give you the same statistics which are already seen in the Excel. But right now we have to merge with the network with our table. So here we can see that there is a node table which contains the shared name and name. So now in shared name, the shared name shows that how one gene is connected to another one. So we will change the name with that shared name. I have to put the same name as a shared name, and the table name same. It must be same. That is after that we will perform the styling process. So after we have imported, and you can see all of the statistics and the shared name are merged with each other. So in styling process, we are going to use the fill color on the basis of log to 4 chain value, and perform the continuous mapping. Here you can see that there is a different color, like red, blue, whitish. So it will give you the idea about the which genes are which gene are upregulated and which gene are downregulated. Here, as an example, you can see that here SOX2 shows the color of red, which is comes under the like 11 point around 10 or 11, and SEC 16B. These are the show that these are downregulated, and SOX is upregulated gene. So these are the by this we can differentiate that which gene are upregulated and which gene are downregulated. And to perform, we cannot analyze this network as a whole. So what we do, we perform the clustering. For that clustering process, we will use the M code. We will use the M code, which is molecular complex detection. It will create a number of clusters which shows are they are highly related, or which have some meaning of the biologically related somehow, like protein complexes, signaling pathway, or co-regulated gene group. This form a cluster of that group. So we will keep as a default setting and analyze the network, current the network. And after doing that, we will see on the left panel, we see that there is a multiple cluster is formed in between these networks. I will show you what is the cluster 1, cluster 2. Please select these here. Here you can see that I have selected the cluster with a high score value, and in this part, these are selected as in yellow. Here, these are the cluster 1, and if I select the cluster 2, these are the cluster 2. Okay, let me see it here. These are the cluster 2. Okay, so here we can see that it has the algorithm that it will group the gene data in a particular cluster. So for the further, this will give you the idea about the like we have to focus on this particular cluster at a time. So after that, we have created a cluster, and then after we have to go for the further downstream processing, which will be we have to extract these cluster from the parental network. So for that, I have selected the cluster here, and go with the file, and as we see the new networks section, here is said that from selected node all edges. After selecting this, the parent network will be getting the background, and we will only see the selected cluster here. Yes, so these are the overlap right now. So we will use the organic layout that will give you the idea about that which gene are available in that group or not. So here we can see that some genes are upregulated. I will told you in the previously I have told you that we are using the upregulated and downregulated gene as a both to perform the network. So here we can classify that blue part shows that the downregulated gene, and the red part shows that the red part shows that they are the upregulated gene. Okay, so these we have performed the we have extract actually we have extracted the net cluster one from that network. For further study, we are going to perform the cytohuba to find out the important top scoring gene. We out of these gene, which one is the top score gene, and we like for that for cytohuba to perform the cytohuba analysis in apps. Either we choose the cytohuba or in the left panel it already showing the cytohuba. Okay, so we have extracted the network. In network section we can see that these are the parent cluster and these are the extracted cluster which have that particular genes. And after selecting the cytoba we are going to select that. Here we can see the name of the stem interaction short 3 PSP fund. In cytoba we are going to select the same network, so that's why we will only work on this, not on the parent network. So after selecting that, we have to calculate the node score. It will give you the idea about that what is the score we are using. Which algorithm going to use the for that score. Here for the node purpose, we are going to use the top 10 node, and these are the multiple algorithms which are available for the analysis. But we are going to use the maximum Select centrality, that is a MCC, to analyze this network, cluster network. And after submitting that, we saw that we are seeing that some of the part is red and yellow. These will show the rank. This will give you the ranking of the gene. Okay, so these are the gene which have shown the on the ranking basis. Uh, these the color will be on the basis of rank, and after that we have to extract this table which have the score all, as well as ranking system, rank of the genes. To as now we are going to save the current rank of the this particular cluster as. This is the MCC. Here, here this is the rank, and this is the scoring that are used to divide on the ranking system of the MCC of the genes, uh, of the genes. Uh, After that, now we have the gene data, and we have the upregulated. Now we have to divide these genes on the basis of upregulation and downregulation. Okay, so here we can see that blue part we have to divide these genes on the basis of current system, because red one is the upregulated and downregulated, blue one is the downregulated. So we have already I have already prepared the table for that, but first I will show you CXCL. Here is no, sorry. So we have to compare this part with this. These are the CD80. CD80 comes here in downregulated. So we will divide these 10 genes in upregulated and downregulated manner. I have already created the genes data set on the divided the data on the basis of upregulated, downregulated. Here, CD80 will be downregulated, and CCR7 will be the upregulated condition. So I have already differentiated that. Then after that, we are going to perform the functional annotation of these genes. So for that functional annotation, we are going to use the tool is sorry, not Cyto. It will be the Plugo. Plugo will give you the idea about the biological and pathway relation with of gene with the like a pathway, give you the relation of the pathway and genes. And here we can see that this panel is open for me. Okay, so I have these genes, upregulated and downregulated conditions. So this we are going to perform the functional analysis of these genes, and for that going to provide the we are going to use the marker list. Marker list will be the our MCC list, and I have created a new column which contains the regulation condition like upregulated or downregulated, and we are going to copy and paste the upregulated condition as a blue section, and the downregulated as a red section. So first we are going to use the downregulated, copying these genes into downregulated and upregulated. So here, I have copy and pasted the upregulated gene and downregulated gene as a different cluster. Here we are going to visualize as a cluster for, and now we have to choose which pathway, I have to find the relation between the of the gene with the biological process, that is the ontological pathway one of the, and the reactome pathway. Okay, so and in next we have to choose the geoterm fusion, and in statistical option we are going to use a custom reference because we are not going to take as a whole gene as a universe. Right now we have only this much of gene, so we are using this gene as a reference for our particular cluster. Okay, so I have created the background gene. Background gene generally it means like we are going to take a pregnant and non both as a background gene, and after that we will start the analysis, global analysis. So it will initialize into the people. This will give you the functional relationship between the gene and the biological process or pathway. Let it complete. After that, we will have the cluster which contains the well, Pluto will why we use the Pluto, like Pluto will give you the enriched analysis of the particular gene and the pathways. Let it complete. Okay, that's here. We can see that these are the particular pathway or biological process system which are involved in the involved in the the gene are involved in these particular pathway or any of the signaling process. So here we have seen that we have already said that these some genes will be the downregulated and some genes are will be the upregulated. Here the red portion is the downregulated condition, and the blue portion shows that these are the genes we are giving. These are the upregulated condition, and these are the downregulated. Means the genes which follow the pathway are in downregulated condition, and okay. So this step is where our network become the biologically interpretable. So we can now link up our the computational findings to back to mechanism of the drug action resistant or disease progression. Finally, we can export the enrichment table. I will show you the enrichment table. These are the enrichment table. Here we can see there is a two cluster, blue and red, and I will cluster one. The cluster one will be the this portion. It involves majorly two things: cytokine signaling immune system and the lymphocyte activation. And in cluster two, there are only like three biological processes involved, like response to cytokine in the response regulating the cell surface. So by here we can extract the cluster data, like functional data of the genes, and by this we can finally export the gene table to visualize the map, and which will serve as the foundation for further study, like further therapeutics insight of the particular disease. So this will be the so as a final what we have done till now, like we have taken the genomics data and constructed the network, and we will use the string for constructing the network, and after that we have found that we will perform the module, we will uh, like perform the M code and set over to find the functional cluster and key regulatory genes. And after that, for functional enrichment, we have performed the biological interpretation. For functional enrichment, logo will be performed to get the gene to biological process and drug response pathways. And this will give you the insight of the hub gene, the enriched pathway guide the biomarker discovery, drug targeting, and the personalized therapy. Okay. So I have completed. So, is there any question?
Thank you, Satya, for the insightful demo.
Okay.
Now it's time for the question and answer season. If you have any questions or
Mhm.
queries, you can ask by raising your hand, tapping in the chat box, or directly you can unmute your mic. So please ask any questions.
Okay, so I will think, I have a question. If somebody has question, they can mail me. Surprise, surprise.
Yes, Satya, any problem?
Yeah, I don't think anyone had question right now, so
Chandan and
they can.
Jishan, please turn off your camera. Chandan and Jishan, please turn off your camera.
Mr. Chandran and Ms. Zisan Fatima, we are requesting please off the camera. I think they are not
Okay,
there.
Maybe Professor Caesar will join shortly. So if you have any discussion query session, like you can interact with Mr. Vivek.
yes,
So you need to wait for some time. Maybe he'll join soon.
okay.
Please don't hesitate in asking the questions. You have a very big opportunity to clear your doubts, to share your ideas. So don't hesitate. So I think there is no questions. So now it's time. Thank you
Thank you,
so
sir.
much, Mr. Satya Vivek Jaiswal,
Thank you,
for
sir,
this
for
informative and engaging demo. We truly appreciate the time and effort you put into sharing your valuable insights with all of us. Thank you. Thank you, Satya. I
thank
am
you, sir, for
glad. I am glad.
Sartoros,
Yes, sir.
Professor Fuente has joined.
Yes, sir. Yes, sir. Yes.
Hi, everyone.
Hi, hello.
Hi, sir.
Hi, great
I'm
to be
glad.
here. Thank you for the Thank you for the invitation.
Yeah. So suppose you can start.
Okay.
I
I'm
can
glad
start. Okay.
I'm glad to share that our honorable speaker, Professor Cesar de la Fuente, has now joined us. But before we begin the season, I would like to invite one of the co-coordinators of this FDP program, Dr. Sintu Kumar Samanta, Associate Professor at IIIT Allahabad, to kindly address the participants and give us a brief and wonderful introduction of Professor Fuente. I would now like to hand it over to Dr. Samanta, sir.
Okay, thank you, Sapros. Professor Cesar de la Fuente is a presidential associate professor at the University of Pennsylvania, where he leads the medicine biology group. He is one of the youngest tenured professor in the history of pain medicine. He completed postdoctoral research at the MIT and earned a PhD from the University of British Columbia. His research goal is to use the power of machines to accelerate discoveries in biology and medicine. Notably, he pioneered the development of the first computer-designed antibiotic with efficacy in animal models, demonstrating the application of AI for antibiotic discovery and helping launch this emerging field. His lab is at the forefront of developing computational methods to mine the world's biological information, leading to the identification of over 1 million new antimicrobial compounds. These efforts started by exploring the human proteome as a source of antibiotic for the first time. His team was also the first to find therapeutic molecules in extinct organisms, launching the field of molecular de-extinction. Furthermore, Professor Fuentes' lab has broadened his antibiotic discovery initiative to explore other branches of the tree of life beyond eukaryotes. By computationally analyzing microbial dark matter, his group has identified nearly 1 million new antibiotic molecules. These molecules have now been made freely available and open access to the scientific community to encourage researchers worldwide to synthesize, characterize, and further develop them. Additionally, through the computational exploration of thousands of human microbiome, De La Fuente and collaborators discovered a myriad of new antimicrobial agents. Collectively, these efforts have dramatically accelerated antimicrobial discovery, reducing the time required to identify preclinical candidates from years to just a few hours. It is estimated that the work of De La Fuente and his collaborators has multiplied the speed of antibiotic discovery by a factor of several millions, saving many years of human research and reducing months to 2 decades of collective work to just few hours. Till now, Professor Fuentes has published over 180 peer-reviewed papers in top-tier journals such as Cell, Science, Nature Microbiology, Cell Host Microbe, Nature Communication, PNS, ACS Nano, and has secured 16609 citation with the H index of 63. Professor Caesar served on the editorial boards of numerous scholarly journals. He has been named as a highly cited researcher by Clarivate multiple times. Professor Caesar has given over 350 invited lectures. Due to his great work, he has received several awards and honors, which sum up to more than 80 major awards. And to name a few, Slogan Fellow 2025, Fleming Prize 2025, Michael S. Brown New Investigator Research Award 2024, Sigma Xi Young Investor Award 2024, ASM Award for Faculty Career Basic Research 2024, 25 Visionaries Shaping the Future of Artificial Intelligence and US 2024, and many more. We are therefore honored to have with us Professor Fuente. With this, I welcome Professor Fuente with a big round of applause. Over to you, Professor Caesar.
Fantastic. Well, thank you so much for that very kind introduction. Can you see my screen?
Yes, it's visible.
Okay, perfect. You can see the full screen now, right?
Yes.
Okay, well, again, thank you for that very kind introduction. It's great to be here amongst this great group. And what I thought I would do is tell you a little bit of the work that we've been doing over, for over more than a decade. We've been working on trying to incorporate computational tools, AI tools, to complex fields like biology, infectious diseases, and antibiotic discovery. And it's quite fun we started a lot of this work before people or useful to apply algorithms to some of these complex fields, because the overall understanding or the notion was that biology was just too complex, too multidimensional for computational methods to, a big impact. I'll tell you some of the progress to make over the years. So for a long time, I've been fascinated by what you're seeing. This is a seemingly very simple chain of letters. That it's a polypeptide chain, it's a peptide chain, right? So we have different amino acids as the building blocks stitched together to form this molecule. And I've always, for a long time, been incredibly curious about this, because peptides are amongst the smallest functional biomolecules from the very beginning of life on Earth. And so if you think about it in that way, if we are capable of understanding how they work from a first principles perspective, the underlying chemistry and the physics, might be able to leverage them to create new technologies that can help us solve a lot of problems that we're facing today. So I believed that for a long time. And so basically everything that I'll show you today is around how we can design and discover these small peptide chains in biological systems. Peptides are actually the ground for AI systems. They have an astronomical sequence space, so that would be impossible to sample through human exploration using just the human brain. And so having a lot of these algorithms is extremely helpful in an endeavor that would otherwise be absolutely impossible. Peptides are small, therefore we can make them. We have these chemical robots where we can do solid phase chemical synthesis, and we can scale up the synthesis in a really high throughput manner. So we can make them in a very reproducible way in the lab. And peptides are incredibly tunable molecules, which you can't say that about many compounds. But by modifying the optimization function of your algorithms, you can actually go after different functions, right? A lot of what I'll present today is around trying to fine-tune or trying to optimize for antimicrobial activity. But peptides can also have anti-cancer properties. They can modulate the immune response. They can enter into human cells. They can travel from organ to organ. So really, they can do incredible things that I believe in the future of peptide design, we might be able to harness a lot of those functions to come up with new technologies, including new therapeutics and so on. And more from a philosophical perspective, perhaps, if you think about the molecules of life, we have DNA, which is really good at storing information. We have RNA, which is a messenger molecule that transfers the information from the DNA to making proteins and peptides. But then we have proteins and peptides, and we don't yet know how to program them from an engineering standpoint, which we do know how to do. We know how to read and write DNA. We know how to now use mRNA to create new therapeutics, new vaccines. But with proteins and peptides, this is an exciting new frontier. And that's what my team is really thrilled about, this possibility of understanding peptide design from first principles and be able to leverage it to benefit humanity and improve the world. And out of all the problems that we could focus on, one of the big thrusts of my lab is to try to come up with solutions to the AMR problem. AMR stands for antimicrobial resistance. I think it's the greatest existential threat to humanity. In fact, if you study history, the number one killer of humans has been infectious diseases throughout. So, and these bacterial infections are currently associated with about 5 million deaths per year around the globe. And the projection, if we don't come up with new solutions, is that by 2050, that number is going to double to 10 million deaths per year. As you can see in this graph, it's projected to surpass every other major society, including even cancer. And this is a big problem all around the world, but actually in India, antimicrobial resistance is a big problem. It causes a lot of deaths. So I think actually in India, having initiatives to fund new antibiotic discovery projects and efforts, I think is going to be really, really important. And I'm really excited because I'm going to India, to Bangalore next month, to an American Society for Microbiology meeting. I think it's the first ASM meeting in India. So I'm really excited to engage with our Indian colleagues and try to come up with solutions together to this big problem. The other thing is that it takes a long time to come up with solutions to this problem, right? It can take up to 6-7 years to discover preclinical candidates or molecules that might be interesting or promising. And it's really expensive. These are US dollars, but it takes over $2 billion to develop some of these antibiotics. So these are some of the gaps, right? Some of the valley of death, right? And so for many years ago, we had this idea that perhaps we could use computers and machines to try to invent new antibiotics, right? And like I mentioned earlier, this was at a time when nobody believed that machine intelligence could be used in the context of a complex field. And in fact, a lot of the efforts were around using algorithms for cell phone applications, like pattern recognition things, like recognizing faces or sounds, right? But we had this perhaps ambitious idea that maybe we could train a machine to come up with a new antibiotic. And we thought about how to do this for a long time, along with my collaborators. And after much thinking, we decided to actually mimic the greatest engine that we have to generate diversity in the world and novelty. And that's, of course, if you think about it, it's evolution itself, right? Evolution has given rise to all of us and all the incredible diversity at the biological and chemical levels that we see around us. So we thought that maybe we could train an algorithm to execute Darwin's theory of natural selection, but do that on the computer, right, in silico. So instead of having to wait for millions of years for a molecule to get evolved to become a better antibiotic, maybe we could do this in a few hours. So just briefly, I'll tell you how we did this. We started with an initial population of small peptides from nature, actually. We encoded them onto the computer, and then we created this program that performed evolution. If you remember from school, the primary steps of evolution are mutation, selection, and recombination. And then we built in this iterative feedback loop so that you get evolution in real time. And this is essentially an evolutionary computation problem. And upon increasing number of iterations, you see how there's an increase in the fitness value that correlates with predicted antibiotic activity. So you can see that, at least theoretically, a lot of these molecules are getting converted into variants, synthetic variants that are predicted to be more active against bacteria. This is all driven by this fitness function here. So of course, this is all algorithmic work, theoretical work. So then we had to do experiments, and we always do the computational work, but we always believe that it's crucial to do experimental ground truth validation to see if our models are working well and how well they're working or how badly they're working. But it gives us a real world sort of measure of how things are going, right? So we synthesize a lot of these optimized synthetic compounds, and next we wanted to see how they actually kill bacteria. What was the underlying mechanism of action? And our expectation was to see this sort of graph here, because previously described peptide antibiotics, the way they kill bacteria is they depolarize the membrane. So we expected to see in these mechanistic assays something like this, right? But what was actually remarkable is that we observed the very opposite effect. So our antibiotic, the lead one that we found, it killed bacteria by doing the reverse, the very opposite, by hyperpolarizing the membrane. And this is the effect that we saw here. So this was really surprising because this mechanism of action was not written into the fitness function, meaning the human programmers that came up with the fitness function did not incorporate anything like this, right? And so I think it's perhaps one of the early examples in complex fields like biology of the emergent properties of algorithms, where that can come up with entirely new concepts. In this case, a new way of killing bacteria, right? We tend to associate imagination, creativity with the human brain. But here we have an example with quite a simple algorithm coming up with something that is actually novel at the biological level, right? So I think that was certainly quite telling and predictive of how a lot of this field might move forward. And then this is the lead compound, guanine-2. We ended up publishing this in 2018, although we started the work about 3 years earlier, I think. And we tested it in a preclinical mouse model. This is a abscess skin infection model where the mice are infected on their backs, and the infection causes this abscess that is really recalcitrant and difficult to treat. And it's caused by Pseudomonas aeruginosa, this gram-negative pathogen. And what we saw in these experiments was actually quite revealing, in the sense that guanine-2, when we treated the mice, we saw a significant reduction in the infection compared to the untreated control group of mice. And this was maybe the aha moment where it really convinced me and my collaborators that this could be an entirely new field, right? Because you could, in this case, use evolutionary computation to design synthetic peptides that not only did they look good on the computer screen, but when we synthesized them, they were actually highly active in vitro. They had a new mechanism of action by which they kill bacteria. But most importantly, I think, they were also active in a mouse model at reducing infections. So since we published this work in 2018, there have been a lot of other groups that have joined these efforts to try to use computational methods and machine intelligence to really try to provide solutions to the mark crisis and to come up with out-of-the-box ways of thinking about this. So this is a lot of the early work that we did. We then decided to ask this. We're really curious about this other big question, I think, which is, can we use machines to accelerate antibiotic discovery? Not only come up with new molecules, but also accelerate the very process of discovering antibiotics. And if you think about it, the way that we've traditionally, in medicine, the way we've traditionally discovered new compounds is it's an extremely physical process, in the sense that scientists go around nature in these expeditions through nature, and we dig into soil samples, we dig into water samples, and we try to then take those very complex samples and purify active compounds, right? Things that might have antimicrobial properties. But as you can imagine, this is a very painstaking process that relies on trial and error, and oftentimes does not yield any exciting candidates at the end of the day, right? So it's a very complicated process. Instead of doing that, we decided to kind of think beyond that and break through that traditional paradigm. And we decided to start from first principles and think of all of biology as an information source, right? As a bunch of code. And if you think about it, DNA is a 4-letter code, and proteins and peptides are a 20-letter code with the 20 amino acids. And if you think of it as just a soup of letters, a bunch of code, we can then come up with algorithms that can mine or explore all of that code digitally to try to identify molecules that might be hidden, functional molecules that might be hidden or encoded within this code, right? So the analogy is, can we develop algorithms that operate like magnifying glasses that zoom into biology and identify new molecules like this one that we see here? And we had the advantage that we have a lot of biological data or information available to us through genomes and proteomes and metagenomes that have been sequenced over the years, right? So that was later work, but that was the impetus for a lot of this new area of work. So I'm going to describe chronologically how we did a lot of this research, and I'll go sort of increasingly in terms of the progress that we've been able to make. We started our early work with great collaborations. We started by mining proteins to see if we could find fragments encrypted or hidden within the proteins that might have antimicrobial properties. So we can visualize a protein in 3 dimensions, like we see here, but also in 2 dimensions, right, with the amino acid code here. And these algorithms, they run through the code and they identify regions that are predicted to be antimicrobial. And these are given by these different colors here, by this scoring function. This is only a prediction, but it's quite powerful because it enables us to identify fragments within proteins, for example, this one in yellow, that are predicted to be a new antibiotic. And then with synthesis, we can extract that fragment and we can try to develop it into a potential therapeutic, right? We did this kind of work for a couple of projects, and we published a number on this, doing 1 protein at a time of the exploration. That was the state of the art, right? But then we realized with advances in compute power that we might be able to scale this up. And maybe instead of just mining 1 protein at a time, maybe we can do this in parallel. We can parallelize this approach and do many proteins at a time and use our algorithms like a barcode scanner that scans through proteins very rapidly and identifies different codes that represent potentially new biofunctional antibiotics. So that idea is what, of scaling the process, is what took us to embark on this project, on this adventure of mining the human proteome for the first time as a source of antibiotics, right? This was an engineering problem in the sense that of the scaling, but also obviously a foundational research project also in the sense of, can we find new molecules in the human body? In the end, we took into account protein isoforms, so we sampled through 42,000 proteins. So imagine, right, we're going from 1 at a time to 42,000. So this was an amazing sort of endeavor, no? in that sense. This enabled us to discover a whole new world of antibiotics that we call encrypted peptides. We found thousands of molecules that had not been previously described in the human body as having antibiotic properties or playing a role in the immune system. So here we are with a very simple algorithm unveiling to the world a myriad of new molecules that we had never seen before, right? This is all computational work, but then we synthesized dozens of these peptides as well. One thing that was remarkable is that out of these thousands of small peptides that we found, 98.3% of them came from non-immune proteins or proteins not traditionally classified as playing a role in immunity. And only 1.7% came from immune, prototypical immunity proteins. So immediately what we thought is that this could be a way of, we came up with this concept of encrypted immunity where there might be hidden layers of small peptides or proteins or metabolites that are not coming or not with traditional immune proteins that might be playing a role in host defense. And so we came up with this idea of encrypted immunity and more broadly encrypted biology. We think that perhaps biology has all these complex layers of encoding that enables it to perform additional complex tasks that get activated, right, upon need, upon context. And we're currently testing this hypothesis of encrypted immunity. I think it's an interesting concept. In experiments in the lab, what we saw is that a lot of these encrypted peptides, they have broad spectrum activity, so they can kill both gram-negative and gram-positive bacteria. Through other methods, including machine learning methods, we've seen that many of these peptides are predicted to be cleaved off by proteases from the parent protein. And the parent protein typically has a completely different function. So it's not the protein does not play a function in the immune system or as an antibiotic, but might play a role in the nervous system, cardiovascular, digestive system. So completely unrelated functions. And these peptides are capable of moonlighting in terms of their function with respect to where they come from. And I already mentioned this hypothesis that we have and this concept of encrypted immunity. You can check out the papers where we propose some of those things. We then went on and saw that these encrypted peptides actually display antibiotic activity in preclinical mouse models. These are two different experiments with Pseudomonas or with Avamani. And in gray, we have the untreated control group of mice, and then we treat them with one peptide, another peptide, or a cocktail of these two peptides in purple, where we see synergy, actually, synergistic interactions in this mouse model. So this combine them in a cocktail together, they actually potentiate each other's activity to target pathogens. They have these synergistic interactions in some cases, some of the pairs. And I think that might be predictive of what might be happening physiologically as well. Maybe a lot of these peptides are produced at once, and then they work in conjunction to target invading pathogens. We went on and we've observed that some of these molecules are produced at different levels, from low to medium to high, in different parts of the. And again, they have a whole new branch of host immunity. And we recently published this paper in Trends in Biotech, where we synthesized a lot more of these encrypted peptides from the human protein. And we saw activity. They also possess, in some cases, very powerful ability to modulate the immune response and reduce inflammation and do things like that. So very exciting to continue to uncover what these peptides might be doing physiologically, and what they might be able to do if we know how to engineer them. The editors of Trends in Biotech, they highlighted this concept of that these peptides might be a hidden layer of immunity that we didn't know about before. So a lot of this work, I really sparked a lot of new ideas in my lab. One of which was, perhaps these encrypted sequences are conserved throughout evolution and not only found in the human proteome. And so we decided to actually explore whether we could find them in our closest ancestors. We are Neanderthal, to see if there was some level of conservation. In the process of thinking about how to do this, we came up with a new framework for identifying and studying molecules over time that we call molecular de-extinction. The ultimate goal of molecular de-extinction is to discover functional biomolecules. These can be small peptides or proteins or metabolites or small molecules, and then be able to synthesize them. In our case, we use solid phase chemical synthesis for small peptides in the lab, so that then we can try to look mutations or changes that occurred in these functional biomolecules throughout evolution influence their biological function, their biological activity, which can be antibiotic properties and ability to influence immunity, or it can be anti-cancer characteristics, or whatever you're interested in. So this is a framework for thinking about how to do this. And we applied it to ancient humans. So in order to sample through ancient human data for new antimicrobials, we developed this machine learning pipeline that we call PanCleave, that at a very high level, it takes all the proteins, let's say from Neanderthals, and it performs computational proteolysis. So it chews up all the proteins into small fragments that are predicted to be encrypted peptide antibiotics. And then we actually run filters, including machine learning, to give us out of those fragments, which are predicted to be the best potential antimicrobials, right? So this very simple pipeline enabled us to identify the therapeutic molecules in ancient biology, including, here we can see encrypted sequences in Denisovan proteins and Neanderthal proteins. And as a control, we use the modern human proteome to sort of have a comparison with the archaic proteomes. But of course, this is only theoretical work, right? A prediction. So then we did synthesis and experimental validation of these small fragments. So in those experiments, subsequent experiments, one thing that I thought I would highlight is that we saw that the modern peptides tend to have a different mechanism of action of targeting bacteria compared to the ancient ones. So the modern ones tend to go after the outer membrane of gram-negative bacterial pathogens, whereas the ancient ones tend to target the cytoplasmic or inner membrane. So I think this is a good example of what might be possible by applying this molecular de-extinction framework, where we can study molecules throughout time, and we can try to learn from them, and that can lead to unlocking new biological insights. For example, these mechanistic differences may be due to evolution, right? Like things that occurred throughout the evolutionary process. And then we tested all of these in preclinical mouse models. This skin abscess model that I mentioned earlier, and then also a deep thigh infection model, which is the one that the FDA in the US, the Food and Drug Administration, typically wants to see for exciting preclinical candidates. And we did a lot of this work in these mouse models, and the best molecule was Neanderthalin 1. I haven't mentioned this, but we had to come up with new names for all these compounds. So we decided to use this convention where we named them based on the organism where they come from, right? So Neanderthalin 1 comes from Neanderthals, right? So this was, I think, a really interesting proof of principle demonstration that we could identify molecules in extinct biological data, potentially therapeutic molecules. And it gave us the courage to ask a more ambitious question, which was, why not biology? Why not just try to and see if we can sort through all of ancient biological data to try to identify new functional antibiotics? When we went to the drawing board to see how we might be able to do this, we actually we encountered another engineering problem in the sense of scaling, because here we're only doing the biological data from 3 organisms, right? We're doing Homo sapiens, or humans, and then Neanderthals and Denisovans. But if we want to do all of ancient biology, that's hundreds of organisms. So this essentially meant that we had to develop a more powerful AI system, and so that's when we started a lot of this big project, coming up with Apex, which is a new AI model. Apex here is the architecture, and we developed this from scratch in my lab, and we trained it from scratch using standardized, experimentally validated data, and that's what we fed into this algorithm to be able to learn how to identify new antimicrobial sequences in biological data. I think the powerful thing about Apex is that you give it an amino acid sequence, any amino acid sequence, and it directly predicts the antibiotic function of that amino acid sequence. So it doesn't take into account structure yet, although we're working on incorporating some structural elements, but it's a sequence to function prediction model, which is arguably actually a harder problem than incorporating structure, because that gives you additional layers of information. Here, from the sequence alone, the model has to predict the function, what the molecule is doing out in the world. Apex was powerful because it enabled us to sample through all of ancient biological data. And here I'm showing some organisms and some peptides that we found in each of those organisms all throughout evolution. Apex took us through a journey through evolutionary history, including the Holocene and the Pleistocene. And here I'm showing representative organisms, some of them where we found the top candidates, the top lead compounds. It includes ancient penguins that were extinct in the 50s, so quite recently. Ancient zebras that disappeared throughout evolution. Magnolia trees that also, stopped or got extinct in the 1800s. Ancient elephants, woolly mammoths, giant sloths, and many other creatures that used to roam around our planet at one point in time, but unfortunately disappeared throughout the process of evolution. And now we have an opportunity to study their biological data, their biological information, and try to extract useful information from these molecular fossils or relics, to see if we can come up with new therapeutics and new solutions to solve present-day problems. So all of ancient biology is captured in this one graph, which took a long time to make, actually. But these are all the ancient biological samples that Apex sampled through. And you'll see that we have both predicted and active. Whenever it says active, we actually synthesize those small peptides and we validated them in the lab experimentally, so doing experiments. So there's a lot of work that is behind this one graph, but it's quite remarkable to be able to see it in just one slide and one graph to see all of ancient biology really captured. It gives you a sense of the scale of the exploration as well. And then we took all this to mouse models, and once again we have on the top this abscess model and the bottom the deep thigh infection model. And we saw promising candidates, particularly from the woolly mammoth, a molecule that we call mammuthusin 2, but also from the ancient sea cow, the giant sloth, and the extinct giant elk. Those were the top candidates. And we, in this case, observed anti-infective efficacy that was comparable to some of the best antibiotics that we have, like polymyxin B, which is a last resort antibiotic. It's only used when absolutely needed. So this tells us that by scaling our exploration to include more biological data, that can give us, of course, molecules that are even more potent, more promising than when you don't mine as much biological data, right? And now what we're doing is we're applying molecular de-extinction to be able to study molecular evolution in relation to its function over time. And here's just one example of something that we're doing at the moment, where in red we see the different mutations that arise over time in a particular peptide molecule. And in parentheses, we have the antimicrobial activity. So how those changes relate to the biological function of each of those molecules. And this is what we're doing a lot now to try to understand molecular diversity over time and sequence function relationships. And I think we have an amazing opportunity now to uncover a lot of the secrets of life, right? And that we really couldn't study before as easily as we can now. In terms of next steps in this field of antibiotics AI, we would like to mine, we think of all of biology as an information source, as we can see here, as a barcode, and we plan to mine all of it for new functional molecules. We're focusing on antibiotics, but in the future, we'd like to do other things like immunomodulators, anti-cancer molecules, and so on. And one of the early dreams that we had was to actually mine the whole tree of life. The tree of life is divided into 3 branches, where we did a lot of the early work in modern and ancient humans. But then you also have archaea and bacteria, which are 2 huge branches of the tree of life. So we wanted to sample them as well. And so I'm going to tell you briefly about each of those projects. In this one, with our collaborator, Luis Pedro Coello, and his lab, we use machine learning to sample through the global microbiome, which are also bacteria, and almost 90,000 microbial genomes that come from all around our planet. So it's a great representation of the microbial life on Earth. And we did all of this digitally. We found remarkably almost 1 million new antimicrobial sequences, which we have released out into the world through this website that we call AMP Sphere. So anybody from anywhere in the world has access to all these molecules to develop them further, to play around with them. We did this in an attempt to accelerate discovery, right? And also to encourage collaboration, right? Worldwide global collaboration to try to target AMR. And then we did a lot of experimental validation. You can check out the paper here if you're interested in more details. The other collaboration with our colleagues at Stanford, Amy Batt and her wonderful lab, we looked at human microbiomes. These are good bacteria that live in and on us, for example, in our mouths, and skin, intestine, urinary tract. And we thought that maybe they were producing also these encrypted peptides that would help them survive against competitors in their surrounding areas, right? Because bacteria, if you think about it, that live in our intestines that are good for us, they still need to sort of grow and propagate in a particular niche. And in order to do that, they need to outcompete competitors, right? So what we saw is, at least in some cases, some of these bacteria produce small peptides that we think they use as a form of chemical warfare to outcompete surrounding microbial competitors, right? So that's how we are thinking about it. And we've taken some of these all the way to mouse models. of infection. And the best molecule that we found, what we call it Prevotellin-2, that comes from the gut microbe Prevotella copri, which a lot of us have in our intestines. So it's quite interesting. I think another example of the remarkable capabilities of microbes to come up with interesting chemistries and interesting solutions that then we humans, by studying them, by learning, can leverage a lot of those things to maybe to treat human infections perhaps. And finally, we just published this paper a couple of weeks ago. We were able to sample through Archaea, the third branch of the tree of life. And Archaea are these really esoteric organisms that for the longest time, scientists thought they were bacteria. But then pioneering work by Carl Woese and others, by sequencing the ribosomal RNA of Archaea, they figured out that they actually, they belong to their own branch of the tree of life, right? And I think these are interesting because they're completely underexplored as a source of therapeutic molecules. And if you think about the antibiotic field, traditionally, basically fungi and bacteria have been exploited as a source of antimicrobials, but not really Archaea at all, almost. And so this is really a systematic exploration digitally of the archaeal data that we have available. And here we actually use a retrained version of Apex to do this work. It's Apex 1.1, really, where we retrained it with more experimental data as part of the training set. This uncovered a new class of antibiotics that we call archaeacines that were quite active in vitro and in mouse models. So you can check it out, the paper. So to summarize this part, basically, discovering antibiotics traditionally takes many years, even preclinical candidates. Now with the methods we've developed, we can do this in a few hours. We can discover hundreds of thousands of candidates, right? And if we really push our resources, we can synthesize them and test them in vitro at least within 1 or 2 weeks. So it's really an amazing playground for a scientist like myself. I've been dreaming about this for a long time. And now we can really do a lot of this work at digital speed, as opposed to taking many years of hard work. And I think the keys here have been not only AI systems and AI algorithms, but also having a lot of really good standardized data that is experimentally generated and vetted, that then we use as part of the training set. And I think us betting on that has been one of the keys of the success of this field. The impact of Apex and some of our other AI models here are some of the things. They've multiplied the speed of discovery by a lot. We've discovered now millions of potential candidates, both in living and ancient biology. Of course, there is no need to culture cells because on the computer, right, by using biological data. This has saved many years of human research time, of course, because we do it with algorithms in silico. And people have estimated that a lot of this work is the equivalent to thousands of PhD students working for 6 years each. So it's quite amazing, right? The world that we live in, where things that, this was impossible 5 years ago. And now this kind of work at this level of speed is actually a reality. So what next? So briefly, I'm going to tell you about a couple of things that are in the pipeline. Is a way of using generative AI to optimize for antibiotic activity. So this uses generative patient optimization. You can start with a template that is not a good antibiotic. And then the objective function is here. You're trying to minimize the MIC, which is the dose required to kill bacteria. So this has a high MIC, so it's not good at killing bacteria. And this model, what it does is it generates new to nature synthetic peptides that are this whole cloud of dots that you see here, that are predicted to be better than the template at killing bacteria. So then we synthesized a lot of these using chemistry, and we saw that 85% of them were able to kill bacteria in vitro, but most importantly, 72% were able to kill bacteria better. This was the objective function, right, to improve the antibiotic activity. This is really interesting, I think, because it allows for optimization. You might have a different template and a different objective function. Let's say you have a small molecule or a peptide that you want to optimize for anti-cancer properties. I think by using this generative patient optimization system and this Apex Go model, you might be able to do something And this translates in vivo as well. So the optimized variants were capable of killing infections more effectively than the templates, also in mouse models. And then the concept of multimodality, I think, is really interesting. We've seen this concept applied to chatbots, but can we design molecules that have more than one beneficial function? If you think about it, every drug that we take in hospitals, they only have one beneficial function, right? Could we change that? Could we design molecules that have more than one? And we decided to apply this concept to a big problem in infectious diseases, which is the problem of intracellular infections, where these are bacteria that are, of course, really smart. They infect us, and then they penetrate into our cells, our human cells, and they hide in there from things like the immune system. So the immune system cannot target them, or antimicrobial therapy. So antibodies cannot really, penetrate into human cells and target them. So we decided to use a generative AI model to design peptides, synthetic peptides that have two functions. They penetrate into human cells, and then they find the intracellular infections that are hiding there, and they kill them. This model created all the synthetic peptides that we then synthesized and validated, and we're able to see that some of them were able to reduce intracellular Staph aureus infections in human keratinocytes, human monocytes. We then tagged them with a fluorophore to be able to visualize how they entered into human cells and targeted the infections in there through microscopy. And we also saw this in animal models. I won't go into all these details, but a number of animal models where we treated them with some of these Turing. We call this Turing of Alan Turing, and we're able to see that they were able to reduce the infections in this preclinical stage. And the last model, and by the way, that we call Apex Duo, this model, because of that dual function. And then Apex Oracle is the last thing I wanted to mention. And really, this is a multimodal model that is end-to-end and bridges understanding from genomics, language, and chemistry. And the input of the model is simply a genome of a pathogen. And from that genome alone, this Apex Oracle model is capable of predicting, out of all the molecules that exist in the world, which ones might be active. And it also has a component that de novo it's capable of generating new molecules, both small molecules and peptides that are predicted to be active against that genome. So in terms of practical applications, I think this is a great way of coming up with molecular solutions against emergent, future pathogens that might come along. If you can think of SARS-CoV-3, SARS-CoV-4, hopefully not, or a new bacterial pathogen that is resistant to a lot of antibiotics, we might be able to come up with solutions almost in real time through models like Apex Oracle. So I'm really excited about the possibilities here. This model, we're in the process of experimentally validating it, so we have to see how well it works, but at least the metrics computationally are really good. So I'm cautiously excited about the potential here. And with that, I'd like to thank lab members, past and present. We have a highly transdisciplinary group, and we attract the best scientists around the world. Our amazing collaborators all around the globe as well for enabling us to do the work we do. And I'll leave you with links to our website, email, and social media, both X and Blue Sky. I'm also on LinkedIn. If anybody wants to connect through any of those avenues to chat science, I would love that. And here's a picture of our lab. And once again, thank you so much for having me. Would love to take some questions. Thank you.
Thank you so much, Professor Fionte, for that truly inspiring and insightful talk. And I have a great message for all the participants. So please remember, the art and science of asking questions is the source of all knowledge. So now it's time to open the floor for questions and discussion. Feel free to ask anything or share your ideas. Please ask any questions.
Hello, Professor Puente.
Hi.
Yeah, hi. Thank you so much for the wonderful talk, as usual.
Thank
So
you.
I have this question that so in all of your antibacterial assays, right? You have used CFU per ml for checking the anti-infectious activity. So, so how is CFU per ml a better indicator than the OD measurement? I mean, of course it is, but why do you prefer it? Because I mean, measuring the absorbance is obviously easier and than the CFU per ml thing, right? So, yeah.
Yeah, sure. Thank you for your kind words. We do both. We typically do absorbance in the assays, but then we also do, in addition to that, we do CFU counts. So we do serial dilutions and we plate those serial dilutions in LD plates or Pseudomonas isolation agar plates, and that is the most accurate way of measuring killing, right? By CFU counts, because you get individual colonies and you can count exactly how many you have. So we typically do both just to have as much information as possible on the experimental side.
Yeah. Thank you so much.
Thank you. Yeah, thanks for the great question.
Hello, I have a small queries. Like when
Sure.
you perform this synergistic interaction between the cocktail of peptides, I guess you have done against Pseudomonas and Acinetobacter baumannii. So my
Yep.
queries is like in my lab also we are working on a couple of target beta lactamase, cell membrane protein. We are designing what antibacterial peptides. My
Right.
query is for targeting any proteins. Should we try these combinations? And is there any rationale of combination? Suppose I'm telling I have orders of 10 peptides. Should we try all possible combination and at a time should we take more than two or only two? What is the rationale behind this energy?
For synergy, I think you can do two-way synergy where you combine two compounds, right? Two peptides or one peptide and one antibiotic, and you can do that in vitro, right? There's this checkerboard assays where you can, in 2 dimensions, you dilute your two compounds.
So even we can
You
do
can
two
look at
peptides
synergistic
instead in
interactions.
checkerboard assay.
You can do more. There's a three order level synergistic assay that is you could do. We've done it before, but it's just more complicated. Typically, people just do combinations of 2 at a time. It's not impossible. We've done. It's just more complicated. You have 3 dimensions on your checkerboard assay, and it's a bit more complicated, but not impossible. Yeah, I think in a biological setting, if you think about it, a lot of these molecules are probably interacting with each other in complex ways. So I think synergistic interactions or additive interactions or even antagonistic interactions might become increasingly important to study, I think, if we want to understand how these things work actually in real life.
So how do you suggest treating any drug-resistant bacteria with peptide plus antibiotics, which is resistant to that?
Yeah, you could do that. So a lot of these peptides, they synergize with conventional ciprofloxacin or things like that. So I think there's a lot of promise in looking at. So a lot of them potentiate drug-resistant bacteria to the drug they're resistant to. Let's say you have a clone that is resistant to ciprofloxacin. Oftentimes, you can find peptide pairs that resensitize that ciprofloxacin-resistant bacterium to ciprofloxacin. So they can actually sort of resuscitate some of those drug-resistant clones, and I think that's an interesting property of peptides as well, that they can do that.
Okay, so whenever you screen with any new set of peptides and you're targeting antibacterial activity against any bacteria, so do you set any range of MIC values? Do you set any range of MIC values?
Oh, you do like a concentration. Yeah, you go from like, let's say 64 micromolar down to,
Oh, so
I don't know,
maximum 64 you keep.
or 32, micromolar, and then you go down because you want to see at the lower concentrations whether you see synergistic interactions.
64. Okay.
Because
And
that's the more is when you see it when they have really low concentrations, because that means if you think clinically how you might translate that. If you require a really low dose of both compounds, the selective pressure towards developing resistance also lowers a lot, right? It gets reduced a lot. So that's what's exciting, I think, to look for those really low doses. I mean, if you can get to nanomolar, that even better, but low micromolar is typically the case for peptides.
Okay. And in one of the slides, you have shown that modern encrypted peptide, they basically target the outer membrane. Correct.
Yeah,
Huh?
some of them do. I mean, it really depends on the sequence, because we found so many that we haven't tested all of them, but it really depends on the sequence. Some of them target the cytoplasmic membrane, some of them target the outer membrane of gram-negatives.
So,
right?
So as because you are very much experienced in this domain, what you suggest targeting a membrane will be more, more effective rather than targeting intercellular any protein, because passing through the cell membrane is also one of the challenging. So what do you suggest?
I think both are super interesting and very much necessary. I think one of the advantages of targeting the membrane is that bacteria take a longer time to develop resistance mechanisms, because typically they need to mutate many genes to overcome that. But I think going after intracellular targets is also crucial. So I think the more people are doing in general and complementary approaches, I think the better for science and progress in this field, to be honest. So if you're seeing and if we can, then the problem will be, can you get those antimicrobial molecules inside bacteria? And there actually peptides can help. You can use them as adjuvants to enable penetration into the cells, into bacterial cells as well. So I think designing different approaches might be solutions for AMR, right?
So if I target any intercellular component, we have to make sure whether it is crossing. We have to make sure we have to check by in silico.
Right. You have to check whether the compound crosses the membrane, and
yes.
you might need to tag it with a fluorophore. That's one way in which you can do that. And then you have to do binding affinity assays to see if it binds to your protein that you're interested in. You can use computational methods. We're working on another AI model that we call Apex Gen that creates peptide binders that bind to specific sites on proteins and interferes with protein-protein interactions. So I didn't have time to mention that one, but we're working on a number of molecular computational tools also to enable us to do things like that.
Okay. I have a last question. See, like now the many papers, not many, few papers are coming up predicting on ultra short peptides, which are 2 to 7 residues long. So
yeah, those are interesting.
How do you think it is very challenging, or people should look for it to these ultra short peptides?
I absolutely think people should look at everything. I mean, whatever you're excited about, you should explore it. I do think the field of peptide design is going to grow exponentially, and I'm incredibly excited for that. If anybody here works on peptide design and is interested in a lot of these things, and you have ideas for collaborations, let me know. We're always interested in collaborating with top people. And so, yeah, I think there's a lot of excitement particularly at the intersection with AI systems, right, and machine learning. So I think there's a huge potential here, and I think actually in India you have a lot of clones that you can try to target with a lot of these systems.
So whenever you look for any candidate lead peptide, so let's say two issues. One thing, whether how much they're antibacterial, how much potent. At the same time, how much they are toxic or hemocompatible. So as a newcomer, which one you should emphasize more, like which is more antibacterial? At the same time, you have to ensure its toxicity and hemocompatibility.
I think you have to balance everything, right? Because if you have something that is really, really active against bacteria, but also highly toxic against human cells, then it's not interesting. So you have to have a sufficiently large therapeutic window, which means the concentration at which you kill bacteria has to be a lot lower than the concentration at which you see any type of toxicity against human cells or red blood cells or so on. By the way, I know that I know it's already time, but if I see other questions in the chat, but you guys can just you can email me or contact me through social media with those questions, and I'll reply, because I've seen a couple of really good questions. I have to go now. I have a meeting that I have to attend. But
thank you so much.
Please don't hesitate to contact me, and thank you for joining the session.
Yeah, sure. Okay, thank you. Have a nice.
There are some questions in
Yeah,
the
shoot
chat
me an
box
email
also.
or send me through LinkedIn or social media.
Okay,
Any
Sarfaraz,
questions you
he's
have.
not busy. He wants to leave now. So if anybody has any queries, please mail him.
Here,
Thank you
here
so
let
much.
me let. Okay, okay. Thank you, guys.
Okay, cheers.
Bye bye.
Bye.
So now it's time to begin our next season, which will be a joint talk by Dr. Anupriya Kumar and Dr. Lucas Iberlin on the topic cryptic pocket detection using enhanced sampling for TERAs. But before we start the season, I think I would once again like to invite one of the co-coordinators of this program, Dr. Sindhu Kumar Samanta, to kindly deliver an insightful introduction of our honorable speakers. I would now like to hand it over to Dr. Samanta sir.
Okay, thank you, Sarfaraz. Dr. Anupriya Kumar, she did her bachelor's in chemistry from Bates Pilani. Then she moved on to a doctorate degree in computational chemistry in South Korea on a full government scholarship during 2005 to 2009. After obtaining a doctorate, she worked at AstraZeneca as a computational chemist in antibacterial research during 2010 to 2013. Then to pursue drug-resistant bacterial research, she moved on to Nagawa University, Japan, initially as a postdoc and then as an assistant professor during 2013 to 2018. After gaining experience in computational drug discovery, she pivoted towards technical account management for scientific software as coordinator and now at OpenAI Cadence Design System in Germany. And Dr. Lucas Eberlin, he did his bachelor's and master degree in chemical biology at TU Dortmund University during 2010 to 2016, then move on to do a doctorate and postdoctoral in chemistry during 2016 to 2022 from the same university, funded by scholarship from the Resolve Cluster of Excellence. His main area of research is the development of statistical mechanical solvation models for quantum chemistry and their application to the investigation of protonation and conformal equilibrium of small molecules. Since 2022, he is an application scientist at OpenAI Cadence Design System Germany with expertise in conformal generation and molecular dynamic simulation for binding free energies. With this, we welcome you, Dr. Anupriya and Dr. Everman, and over to you both.
Thank you for the nice introductions, and thank you very much for the opportunity for inviting us. And today we'll be providing a very scientific talk starting from simulations, what is the application of simulation, and how can you detect cryptic pockets in novel proteins or already existing proteins for allosteric drug discovery. And today, because Lucas is an expert in molecular simulations, he'll be delivering the presentation. And this will be approximately for 1 hour. And last 30 minutes, please feel free to ask any questions to us. If we cannot answer today, we will reply to you in the email. So let's get started. I hope you enjoy the webinar.
Yes, thank you for having us. I hope you can see my screen
Yes, sir. Yes, sir. Your
and,
screen
and
is
hear me.
fine.
Does it work well?
Yes.
Thank you. Yeah, thank you again for the very nice introduction. I'm excited to be here and speak to you today, especially since it's already kind of late for most of you folks compared to us here in Germany. So thank you for staying and listening to us. The title of my talk is Drugging the Undruggable, because I'm going to introduce our method for detecting and ranking cryptic pockets to you, which we think is highly accurate. And I will present you some validation studies to underline this opinion. So to start with, here's the outline of my presentation today. I will start by introducing the KRAS system, an oncogenic protein long time considered undruggable to you, and show how cryptic pocket detection can speed up drug design drastically. We will discuss if and how we can use molecular dynamics simulations for cryptic pocket detection and focus on the weighted ensemble path sampling strategy, an enhanced sampling strategy that allows us to run our calculations more efficiently. This will lead us to our OpenEye's solution for the protein sampling problem, and subsequently to our method to detect cryptic pockets. We will have a larger scale validation study to show if and how well these methods work. And I would like to finish the talk by introducing our ligandability model to you. Such a model is important to be able to not only identify cryptic pockets, but also assess their ligandability to really find the pockets you can use for your structure-based design campaigns. Okay. Let's start with the KRAS system. Many of you likely know this system, so I'm just going to give a little bit of background in case you aren't familiar. KRAS was discovered in the 1960s, so the gene for it, and it acts as a regulatory switch by switching between the GTP-bound active and the GDP-bound inactive state. However, there are some oncogenic mutations, especially around glycine 12, that keep it in the on state for its entire lifetime. And this has many, many effects. Most notably, it causes tumors to grow. KRAS is one of the most commonly mutated oncogenes in solid tumors, with KRAS mutations being present in as many as like 20% of such tumors. The prevalence varies widely depending on the tumor type. For example, they are present in 85 to 90% of pancreatic ductal adenocarcinomas. So it really depends on the cancer type. And locking the protein in the off state is a promising strategy for cancer treatment. I already said KRAS was discovered in the early 1960s via the gene, but it wasn't until the 1980s, early 80s, that it was found to be a very important target for cancer therapy. But it was considered an undruggable target for a very long time. It took around 30 years before people would find the switch 2 pocket here in this area of the protein in KRAS, which was discovered by work from Kirwan Shokat's lab at UCSF in 2013, where he was doing some covalent fragment screening and found that this switch 2 pocket was potentially suitable for therapeutic design. And just 10 years later, we now have 2 FDA-approved drugs on the market, sotorasib and adagrasib. And both of these bind to that newly discovered switch 2 site, and several other compounds are in the clinic. So if you think about the story of KRAS, the rate-limiting step for developing these therapies wasn't to find the actual drug-like molecule, it was to find the pocket. So what we would like to do at OpenEye is to remove this 30-year wait and to allow you to find this pocket basically with the push of a button. So there are a few questions that we are asking in order to answer the question whether or not we can develop a solution for detecting and ranking cryptic pockets. And these questions start out with, where might a ligand bind? Given a protein molecule, the simplest question is, on this protein, where are the possible sites where a ligand could potentially bind? And then you refine the question down to, where would a drug-like molecule typically bind? And this is really where you want to start a structure-based drug design campaign, using a pocket where you know a molecule would actually bind to. And finally, what happens if a molecule binds there? So some biological effect. And if we go ahead and start at the beginning, we have to have a crystal structure for any type of structure-based drug design. Usually it's an X-ray structure, but this can be really limiting. And we think that you can be trapped by the structure. And the reason why is that proteins actually move. And so I want to show you this quote from Galileo when he was held for heresy by the Catholic Church for supporting Copernicus's idea of heliocentricity, and yet it moves. So, and we need to take this statement to structure-based drug design. We can't be trapped by a crystal structure. These proteins move around, and we have to move them to see what they actually do. And you can do that by using molecular dynamics simulations. And it might take quite a lot of MD, or perhaps you have another way of doing it, and you can use slightly less MD and use something like a chemical probe. And yeah, we have ideas about how to do that. Now, once you've done some MD, you have to observe pockets, but that only tells you about where might a ligand bind. And now you have to actually rank the pockets to be able to tell someone that this is a really good pocket to start your structure-based design campaign. Sorry? Okay. So let's go ahead and try to answer the first question. How can we come up with a solution to the question, where might a ligand bind? In order to do that, we've been thinking hard about types of motion associated with cryptic pocket formation. Some of them are relatively simple, like sidechain motions, which happen in the order of nanoseconds, where you have a simple sidechain that need to move out in order for a pocket to open, or you have larger motions like loop motions, where you have an entire loop that needs to move out of the way in order for a pocket to open, or perhaps even the reverse process where a loop has to come back on top of a pocket to form basically a lid. Alternatively, there are longer timescale motions like secondary structure motions, where you have to move an entire secondary structure out of the way or bring it on top like a lid. And this is already a little bit more time consuming with microseconds to seconds, and outside of the scope of typical simulations. There are even more complicated scenarios where you have a secondary structure that needs to change, and you go from an ordered to a disordered state, or maybe the reverse process, or maybe even really long time scale motions like interdomain motions, where you have two parts of a protein that may be tethered or not, and they either have to come apart or move together to expose a pocket. And finally, there's another type of pocket that some people consider to not really be an actual cryptic pocket, but nevertheless, they are still pockets that need to be detected and ranked. And these are pre-existing pockets inside your protein. So we think about something like little holes inside the protein, like in Swiss cheese. And these are the types of motions that and pockets that we want to be able to detect. Before I show you how we actually do so, I want to talk a little bit about MD simulations, the timescales we can cover with them, and how it fits to the aforementioned timescales of cryptic pocket opening. So, what is an MD simulation? It's the simulation and analysis of the physical movements of atoms and molecules. We can observe the dynamic evolution of the system over time by applying Newton's laws of motion to the system at an atomic level. Most of the time, molecular mechanics force fields are used as the potential energy functions for MD simulations, but in principle, a variety of different potential energy functions, for example, derived from quantum mechanics, neural network models, or even hybrid approaches are possible. And what is really important, if we sample long enough, we can determine our system's macroscopic properties from such a MD simulation. Sorry. MD simulations are frequently used in drug design. We can use them to explore a huge variety of our system's properties with the prediction of cryptic pockets or pockets in general, and binding affinities probably being the main areas of application. But there are many more things for which MD can be an extremely helpful tool. Today, of course, we will focus on the detection of cryptic pockets. So let's go back to the timescales for even this cryptic pocket formation. While there are some motions like sidechain rearrangements, which are in the order of nanoseconds, most of these motions are in the microsecond to second range, with some going up to minutes. And here on the bottom, we have the wall clock time required to generate MD trajectories of a certain time with a typical desktop computer. We see that it takes a lot of compute and time to even simulate microseconds. Of course, we can speed things up by using proper high-performance computing infrastructure instead of desktop machines, but still, events that happen on the timescale of seconds to minutes can't be simulated with standard brute force MD. Even if it would be possible to run such a long MD simulation, it would not be guaranteed that the event we are looking for actually takes place, especially if the cryptic pocket opening is energetically penalized. To illustrate this, let's have a look at this alanine dipeptide example system. So here we have the free energy surface from multiple, many microseconds of biased MD, so replica exchange MD, and that's a lot of simulation needed to sample such a simple free energy surface, just the rotation around the phi and psi angles of this alanine dipeptide to identify this metastable state here. So we can imagine that for proteins, which consist of hundreds of amino acids, we definitely need methods to speed up the sampling process to be able to observe such slower motions. So again, sorry, here's the metastable state, and here we have the global minimum. One of the techniques we can use is coarse graining. Here, we have the time and length scale associated with motions important for drug design. We can again see that all atomic simulations will not help us to go to the timescales needed for some cryptic pockets to open. Coarse graining is a process of simplifying complex systems by representing them at a higher, more abstract level, focusing on large-scale behavior rather than the individual components. For example, by grouping several atoms of a chemical group into a single effective group. And this helps especially on the length scale, but slower protein motions are still hard to sample. So coarse graining can contribute to, but is not the full solution to properly sample the protein motions needed for cryptic pockets to open. We need to do smarter sampling as well. And there is a variety of enhanced sampling methods out there. It's important to carefully choose the appropriate method for each problem we want to solve using MD, and things like this decision tree can help us doing so. Some of these enhanced sampling methods use bias potential, as shown in the alanine dipeptide study, but we have decided to use a method that does not need to use such bias potentials and allows us to sample the protein conformational space efficiently. Before we get into the details of this method, I want to show you what we ideally want to see out of an MD simulation. Here we have the KRAS protein, and we will see the FDA-approved drug adagrasib. Let's have a look at the trajectory and focus on this region here on the right-hand side of the protein. We see a lot of conformational changes in the protein, including the opening of the switch II pocket and the drug binding event. So how are we able to simulate such a binding event? The method we apply at OpenEye is the weighted ensemble algorithm, which I'd like to briefly introduce to you. This is the free energy profile along an arbitrary progress coordinate x. We have two minima And yeah, we can bin the free energy profile between those minima. We start with a simulation with a single stochastic walker, so an unbiased MD simulation, and run it for a very short time and check if it moved forward along our progress coordinate. Here it hasn't, and we split the trajectories to increase the chance of a simulation to reach the second bin. Our goal here is to always have 2 walkers in each of the bins. And it is important to keep track of the weights here. In this case, having 2